to create complex patient-specific structures using 3D printing and bioprinting opens up unprecedented possibilities for personalized medical treatments and advancements in healthcare. Biomaterials specifically designed for 3D printing, or Bioinks for bioprinting, enables the fabrication of intricate biocompatible structures with precise mechanical and biological properties. By tailoring biomaterial formulations to mimic the characteristics of native tissue, researchers and clinicians can create scaffolds, implants, and organ constructs that promote cell growth, tissue regeneration, and integration with the host body. Moreover, the continuous exploration and refinement of biomaterials for 3D printing contribute to the development of innovative medical devices, drug delivery systems, and surgical tools. As biomaterial development progresses, it drives the expansion of 3D printing. I reiterate, biomaterial development drives the expansion of 3D printing and the industry capabilities, which allows for new solutions for personalized healthcare and revolutionizing patient care and treatment outcomes. So in this upcoming event, we have a number of panelists, experts in this field that are going to be speaking about the current status, immediate future, and long-term outlook on biomaterials and Bioink for healthcare 3D printing. So we thank everyone again for joining. And with that being said, I would like to call the first speaker to the stage, Gurcharan Chowdhury. Come on up to our virtual stage. Very good. Gurcharan,
awesome.
Say hello just to make sure that your audio is okay.
Yep. Hello. Thanks for the introduction, Craig, and thanks for joining us, everyone. I'm excited to be here.
Very good. So Gurcharan, why don't you pull up your first slide and then I'll formally introduce you. Someone has to be lead off. Very good.
Yep. I think you should be able to see it now.
Yes, perfect. So Gurcharan Chowdhury is a polymer physicist and co-founder of 3D Biofiber, a pioneering biotechnology company at the forefront of biomaterial innovation and manufacturing. Is the director of business development and operations. He has raised $5 million in funding within 3 years, enabling the successful commercialization of 5 groundbreaking biomaterial products through direct sales and strategic partnerships. As an innovative leader, Gurcharan is determined to bring 3D biofiber to the forefront of biomaterial fabrication and help move 3D bioprinting technologies towards clinical applications.
Awesome.
Gurcharan, feel free to take it away.
Awesome. Thank you so much for that introduction, Craig. As Craig iterated, my name is Gurcharan Chowdhury. I am the Director of Business Development and co-founder of 3D Biofiber. 3D Biofiber, just as a high level, is a protein fiber manufacturing company. So we're a company focused on making protein fibers accessible in tissue engineering, and a specific focus of ours is on 3D bioprinting, which I'll focus on today. So 3D bioprinting is probably something you've heard of. It's an excellent research tool with great clinical potential. This is where the industry is today. As Craig alluded to in his intro, this is really using some of the key principles from the 3D printing we're all familiar with, but applying it with different materials to print ideally functional tissue constructs that can be implanted back into a human being. But today it's very much a research market where we're starting to see the first examples of 3D bioprinted technologies moving towards preclinical or clinical trials, mainly in diagnostics. But these examples tend to be the exception and not the rule. Generally, the industry is still fairly nascent, moving towards really achieving that clinical potential. And this image I have in the back here is actually from Dr. Feinberg at Carnegie Mellon University, where they've been able to print from an MRI picture a full collagen construct of a heart. So a full-size heart without any of the cells on it, which is quite a bit of a feat, but again, is really moving the research towards that clinical potential of the industry. And in order to achieve that full clinical potential, there are some key challenges to overcome. The main challenges are, one, the low mechanical durability. Right now, when you print a construct from a 3D bioprinting technology, that construct tends to lose its shape over time. mainly because the cells living within it will pull on that construct, causing it to contract. And as it contracts, it becomes too dense for the cells. So they start to die off, and that loss in shape is critical to losing that functionality over time. Another challenge is actually the cell concentration. So in Bioinks, we have cell concentrations that, although sound high and hundreds of thousands of cells per milliliter, in natural tissue, there are more like hundreds of millions of cells in that same unit. So we really need to find ways to increase that cellular content to make functional tissue constructs. And in solving these challenges, there are some features of bioprinting that make it exciting that we need to maintain. The first is this Bioink, which is composed usually of cells and proteins, needs to remain printable. So you have to have the right viscosity to actually be able to print a construct from the hardware you have access to. Secondly, what you print needs to be physiologically relevant. So while you could fix this problem by using things like a plastic isn't very physiologically relevant, so it's not an environment the cells will thrive in. And lastly, the material has to be implantable. So you have to be able to take your solution and put it into a human, with the outcome being success in that integration. And in trying to solve this, really the main challenge is that low mechanical durability. As I was saying, a key part of why these constructs fall apart is actually because of how the cells behave. So you can't increase that cell content until you increase that mechanical durability. So I will argue that the challenge that's really holding 3D bioprinting back from clinical applications is the low mechanical durability of 3D bioprinted constructs. And this has been a big focus of the industry and researchers to solve. And common solutions in trying to target this low mechanical durability are to increase the protein content within the Bioink, because if you have more material, it tends to be stronger. But the challenge here becomes that the Bioink becomes more viscous, so it's too difficult to print. Another option is that you can increase the amount of cross-linking you apply, which is how the protein sort of sticks together to make that final construct. But as the cross-linking gets too high, the construct becomes too stiff, which starts to kill off the cells, limiting the physiological relevance. And something that's on the newer side is the idea of using additives. So using things like rebar and concrete, but using biological additives or synthetic additives to drive the mechanical durability. And one example of that is cellulose, which is very abundant in nature, coming from plants, but it's not in humans. So it's not physiologically relevant to a human cell. So the need of the industry, generally speaking, is to improve the mechanical durability of these constructs while maintaining the features that make bioprinting attractive. And for that, I argue that collagen fibers are actually the ideal additive. Industry has shown with other synthetic additives and things like cellulose that using this rebar and concrete type analogy, adding fibers to Bioinks, does improve their mechanical durability. And collagen fibers are the most abundant protein structure in the human body. They naturally play a key role in maintaining the structure of our tissues, and they're highly physiologically relevant in terms of their definition in the extracellular matrix. But the challenge for industry is that we don't have a reliable supply of collagen fibers. The only thing that makes collagen fibers are animals, and in extracting collagen from animals, we usually have to break these fibers apart into their subunits that are shown here, with no way of stitching them back into a fiber. So there isn't a way to access these materials for bioprinting at commercial scales. This is really the problem that we've solved at 3D Biofiber. We have a proprietary dry spinning technique that allows us to spin protein fibers at commercial scales that closely match the natural properties of that protein fiber. The way that I explain the process is if you think about a spider spinning a web, which is another example of a protein fiber, it involves a spider crawling across the ceiling, depositing some solution, and dropping itself down. That dropping action aligns the proteins within the solution, and that alignment creates an environment for self-assembly. We do the same thing in our process. We start with a spinning solution that's made of 3 ingredients. It's a collagen, a scaffolding polymer, which enables our process and can be removed from the final fiber, and a solvent. We mix these together into a very viscous solution, and then as shown in this video, we contact that solution with a pin array. remove that pin array, and each pin forms a collagen fiber. We have a fully automated technology to produce these, as shown here, where we have a machine that can pull these fibers and lay them down on a substrate, allowing us to make collagen fibers at scale and even create controlled three-dimensional structures using these fibers. And while we're not the first to make collagen fibers, our technology is the first to make collagen fibers at scale that closely match the biomechanical and biochemical properties of natural collagen fibers. And while I won't get into these other techniques of wet spinning and electrospinning that are commonly used for protein fiber production, our technique at 30 Biofibers make collagen fibers that match or exceed their strength in the body, where their main role is to provide structural support to tissues. We can produce these fibers at over 3,600 times the rate of combined technology. So electrospinning can produce about 1,000 meters per hour. In our technology, we readily make 1,000 meters a second. So at least 3,600 times that. And lastly, a key role of collagen is to provide a site for cell attachment. And in order for cells to attach, the diameter of the collagen fiber is critical. So in the body, collagen fibers are 1 to 5 microns in diameter, and we can produce that size very consistently. Whereas wet spinning is an order of magnitude too high, about 60 microns is the lowest it can make. And in electrospinning, it's an order of magnitude too low. So 0.2 microns is about the maximum it can make. So our technology is the first to make fibers of collagen that match their natural properties at commercial scales. And using this technique, we've created a collagen fiber additive, microcollafiber is what we call it, to reinforce bioinks that are commercially available or that are built in-house by researchers. The general idea of this solution is to combine our short fibers of collagen into off-the-shelf inks and having those fibers act like rebar to increase the strength of the final construct, increasing its durability, while also using a natural representative of the extracellular matrix to improve the physiological relevance. This here is just a picture of what the product looks like. That thing that looks like a cotton ball is actually thousands of fibers that are about 50 microns in length that an end user can resuspend in any bioink of their choosing. And then as shown in the schematic, they would have a bioink that has their cells, our fibers, and the general protein that they normally use, resulting in a bioprinted scaffold that is reinforced by our fibers, improving that mechanical durability over time. And this is a project that we've been working on that's been quite successful. So I'll go over some of the high-level data about how we're really solving this challenge for the industry. The first is the mechanical durability. When I started this, I was saying the main challenge is that constructs lose their shape over time. In this example, we've used on the left side here a construct of gelatin methacrylate, commonly called GELMA, which is a very common bioink that's used by the industry. And in this left image, we've seeded it with fibroblasts, just a type of cell, and you'll see that within 6 days, this construct has contracted by 25%. So on day 0, it actually filled this circle completely, and now it's lost 25% of its shape because the cells pull on the scaffold. It can't deal with that force. It becomes more dense, and as it becomes more dense, the cell viability dies off. And that's only 6 days into culture. When we add our material into this, we can see that after 28 days, we actually retain 92% of the shape. So we're really solving that mechanical durability challenge, which allows us to keep over 90% of these cells alive throughout this time, really driving that ability to grow these constructs to reach their full maturity. A second key consideration was maintaining the printability. So what we've been able to show is that the viscosity of bioinks is virtually unchanged with the use of our material. So in the top graph here, on the left side here, you'll see the viscosity of different inks. And on the bottom is a shear rate, which you can take as an analog to flow rate. So a lot of materials tend to become less viscous as they flow, which is commonly known as shear thinning. In this example, we have 5 conditions. The black lines at the bottom is our GELMA only control. Then we have 2 blue lines, which are the GELMA with our collagen fibers at 2 different concentrations. And these 2 green lines are the same GELMA with cellulose nanofibers at 2 different concentrations. And what you'll notice, especially in the red box, which is where most bioprinters operate, that with our fibers, that rheology, viscosity, and printability is essentially completely maintained. Whereas when you use other additives, it can be increased by 2 or even 4 times. really making it difficult for end users to integrate that without having to redefine their entire printing protocols. And then when we look at the load, so the overall strength, our material in blue does almost as well, if not better, than the cellulose does. So we're able to maintain that viscosity while really driving that mechanical durability. The other part of this that I was speaking to at the start was the physiological relevance. So how do cells interact with your material? Here are two examples of that. In the top row on the left side, this is our control of alginate, which is a carbohydrate, a material that cells don't attach to, and fibroblasts growing in them. And what we see here is these fibroblasts have very rounded phenotypes, meaning while they're alive, they're not really doing anything functionally. So they're not very useful as a cell in this system. Whereas when we add our fibers, you'll notice that those cells are much more elongated. They're proliferating. That's why they're clusters of these cells that have replicated out, really driving their functionality. And fibroblasts are fairly robust cell types, but we've seen the same response with very sensitive cells. So in the bottom here, the condition on the left and right are using human mesenchymal stem cells or signaling cells. And what you'll notice on the left again is they have that characteristic rounded phenotype of a non-functional cell. But with those fibers, these MSCs are very happy to attach to them, align, and and become a lot more functional as is required for growing MSCs in a bioprinting application. And lastly, something that we can do is really drive the shape fidelity right after print. So your ability to create a well-defined structure without relying so heavily on cross-linking. So in this example, we're using a commercially available printer from Aspect Biosystems. And what you'll notice at the top is these two prints that have decent shape fidelity actually have a fairly high cross-linker rate. So the CXL is the cross-linker rate and the M is the material rate. So here we're using material and cross-linker rates that are about equal, meaning that you're really heavily cross-linking that material. Whereas when we add our fibers, we're able to get a better defined structure with a significantly lower cross-linking to material ratio. So you're able to push more material out, reduce the level of cross-linking that's required, and improve that shape fidelity, which really lets your cells and your end user tune what the stiffness is correctly to match the cells, because each cell requires a different stiffness. And if you have something that's too stiff, it really doesn't represent their natural cellular environment. So just to summarize what we've been able to show, our collagen fibers really have an ideal additive behavior in Bioinks. Not only do they solve that mechanical durability problem, they do so in a way that maintains the printability of the Bioinks for end users, so they don't have to develop new protocols. It improves the physiological relevance, allowing cells to really grow on materials that represent what they would naturally grow in. And very importantly, it allows people to decouple their local construct stiffness from their overall durability. So you can not only have a construct that's durable, but you can also create a mechanical environment that is suitable for the specific cell type and application you're working on. And we're able to do this by creating an additive that's really representative of the natural cell environment. So that I'd like to thank everyone for their time. That was just a highlight of the product, the collagen, and how we're integrating that into bioprinting applications. If you'd like to connect with me offline, my email's on this site. And over the next 6 weeks, we're actually traveling for a number of conferences, including BioFuture, Cell and Gene, BMES, and Cellink. So if you're at any of these and would like to connect, send me an email or a note, and I'd love to touch base. I'll stop there, and I'm happy to take any questions.
Thank you very much. For those of you that have questions, feel free to enter them in the Q&A box. I'll start off. I have a question. Just understanding the technology certainly is quite remarkable, and I thought your point was certainly well taken as far as the low mechanical durability and ways to overcome that. If you go back to slide 8, I thought that it was quite interesting. Not only does the area retention percentage increase, but also the margin for error, your error bar is significantly reduced. Could you speak to the consistency from batch to batch? Because I know that's usually a concern in this field.
Yeah, a big part of this challenge, especially when you see contraction, is actually how the cells deal with the material. So what this contraction is driven by is that the cells, as they move around inside the gel, pull on that gel. And that pulling action, because it doesn't have anything to resist, it starts to contract. And then that contraction leads to an increase in density, which leads to cell death. So depending on sort of how quickly it's contracting will determine how many cells are dying. And therefore, as cells die, they can pull on it less, right? So when you have this sort of uncontrolled cell death over the first few days, different constructs where cells might be more viable are contracting more, whereas where they're dying quicker are contracting a little bit less. So that uncontrolled cell death really deals with that variability. Whereas in this condition where it can maintain that shape, those cells are alive and proliferating. And that's actually why you see a lot more of these dark spots throughout the construct. These are sort of local areas where cells have proliferated and replicated. And because of that, you're not only able to increase that cell count, but you're able to do so in a way that's more consistent between replicates, as you pointed out.
Very good. Anyone that has questions, feel free to enter them in the Q&A box. Continue to include your own information if you'd like to network with folks. I think that's a great way to make great use of this presentation. I do have one other question. You presented
please.
obviously all of the technical content, but I find your position as Director of Business Development quite interesting, right? So looking at things from the commercial side, it seems like you guys attend quite a few conferences. What are your tactics for being able to raise so much funding in a short period of time and be able to kind of convince other people that your technology is the future?
Yeah, so a big part of it is baked into the business model. So at the core of our business is this technology that is the ability to make collagen fibers at scale. And collagen fibers, being the most abundant protein structure in the human body, have a very wide range of applications across tissue engineering. We know at 3D Biofiber that if we went after every opportunity that we had an application or potential application in, we wouldn't get anywhere because it would be too many shiny objects to go after. So the way that we think about our business is that we are focused on what we're good at, which is manufacturing collagen fibers at scale. And we're going to continue to invest in that to improve our efficiency, our consistency, and our versatility in that process. And then we're looking to partner with the industry as broadly as possible across tissue engineering, where our material can improve their application. So we are working in unregulated spaces, for example, in Bioinks for research, where we've signed partnerships where we're supplying that material to commercial Bioink developers for them to make new inks for their customers, for them to realize some of the benefit that I've been talking about. But we're also signing deals in regulated applications. For example, we recently signed a deal in a nerve repair space where there's a company that's creating nerve sheets for nerve repair. So they wrap a severed nerve, allowing the nerve to regrow. And there might be some applications where, because our fibers actually drive cell migration, we can increase the rate of that healing. So because of that, like, we don't know enough about nerve biology in our team. It's not where our expertise lie, but we know a lot about collagen fiber manufacturing. So we partner with groups that are experts in that end application, like nerve repair, and then together hope to bring a superior product for patients that can realize that benefit.
Very good. Well, thank you very much. I think we all found your presentation quite insightful. It looks like there is one more Q&A, one more question, if you could answer this quickly before we continue to Julian. The question is, can you please share with us, from a level of interest of your customers, what organ or other application will be the first widespread use of your fibers?
Yeah, that's a great question. Part of what we're seeing is that because collagen fibers, as I'm saying, are so abundant in the body, there's really a diversity of those applications that we're seeing. And all organs are pretty complex. We can argue about which organs are simpler than other organs, but the reality of it is every organ is fairly complex. But the areas that we're seeing uptake are in skin for sure. Now we're seeing some nerve applications, which, although not an organ on its own, is an application in a tissue type that we're seeing. But we're also starting to see indications in muscle because of the alignment feature, and for the same reason in cardiac tissue, so in heart, because those cells are so well aligned and our fibers really drive that alignment. Tissues that benefit from that strong alignment is where we're seeing a lot of application.
Very good. Well, thank you for starting things off. We all appreciate your time. And we're going to call the next speaker.